Good morning, children. In the previous session, we have studied about prokaryotic as well as eukaryotic transcription. Now let us learn about the differences between prokaryotic and eukaryotic transcription. In prokaryotes, only one enzyme, that is DNA-dependent RNA polymerase, transcribes all the 3 RNAs. But in eukaryote, 3 different enzymes are involved. DNA-dependent RNA polymerase 1 transcribes rRNA. DNA-dependent RNA polymerase 2 transcribes mRNA but in an immature form as hnRNA. DNA-dependent RNA polymerase 3 transcribes tRNA. And second difference is in prokaryotes there is no split gene arrangement, but in eukaryotes split gene arrangement is seen. What is a split gene arrangement? That is, DNA of an eukaryote has both exons and introns intermingled with each other. Exons are the expressing sequences and introns are the intervening sequences. So during transcription, all the introns are removed and exons are joined by means of splicing process. So in case of eukaryote, split gene arrangement is seen and this leads to splicing process. Third difference, in prokaryotes, mRNA is formed in the matured condition. So there is no capping and tailing. But in case of eukaryotes, capping and tailing occurs. You know what is capping? It is the addition of methyl guanosine triphosphate at the 5' end of hnRNA. And tailing is the addition of polyadenylate residue at the 3' end of hnRNA. So these are the major differences between prokaryotic and eukaryotic transcription. Now next one is about the genetic codon. So you know very well that DNA is the storehouse of genetic information. So how the informations are stored in a DNA molecule. So what is a genetic codon? Genetic codon is a sequence of 3 nitrogenous bases or 3 nucleotides of a DNA or RNA, and that will correspond to a specific type of amino acid. Try to understand here. A genetic codon is a sequence of how many bases? 3 nitrogenous bases. And these 3 nitrogenous bases, the sequence of 3 nitrogenous bases in a DNA or in an RNA code for a specific amino acid. That is what is called a genetic codon. It is a sequence of 3 bases present in DNA or RNA that code for a specific amino acid. These codons either can start the process of protein synthesis or it can stop the process of protein synthesis. Either it can start or it can stop also. Now the question is, if you take a DNA molecule, it consists of, repeatedly it consists of how many bases? 4 bases are there. What are the 4 bases it is having? Adenine, thymine, guanine and cytosine. So if you take an RNA, instead of thymine, uracil is present. So it consists of 4 nitrogenous bases. And what is the function of the genetic codon? It can either start or it can stop the protein synthesis. That means it is playing a very vital role in protein synthesis. So how many naturally occurring amino acids are there? There are 20 naturally occurring amino acids. There are 4 nitrogenous bases and 20 naturally occurring amino acids. So if a codon consists of 2 bases, 4 to the power 2, it can code for how many amino acids? Only 16. So if you take 4 to the power 3, then it can code for how many? 64 it can code. But naturally occurring amino acids are 20. So a scientist, George Gamow, first informed that a codon consists of 3 bases. If you take 4 to the power 2, it can code for only 16 amino acids. But naturally occurring amino acids are 20. So what he considered? He considered the genetic code consists of 3 bases. So how many codons are possible? 64 codons are possible. And what information this codon is having, that is being deciphered by another scientist, Marcel Nirenberg. Now try to understand. Genetic codon is a sequence of 3 nitrogenous bases present in DNA or RNA, and that is specific to a particular amino acid. It can either start protein synthesis or it can stop the protein synthesis. There are 4 nitrogenous bases, but naturally occurring amino acids are how many? 20. So George Gamow was a scientist. He proved that every time if you are taking 4 to the power 3, then 64 codons are possible. And the information what each codon is carrying is being deciphered by another scientist, Marcel Nirenberg. Now you just take here what is given. Base sequence is given present in an RNA molecule. Uracil, cytosine, adenine and guanine. And here what is given? This is the first position, second position and third position of the base. So based on that, all the 64 codons are being shown here, and everywhere you can see that codon is a triplet. You need not by heart all the 64 codons, but you should know few codons very clearly. So what are the codons you should know? One is about the start codon you should know, that is of AUG. AUG. This is called the initiating codon or start codon. This you have to remember. Another one is UAG. This is called a stop codon. This is start codon. This is of stop codon. And one more you remember, triple U. This is for phenylalanine, an amino acid phenylalanine. So 3 codons you should be knowing. AUG, it is a start codon or initiator codon, and then comes UAG, UAA and UGA. These 3 are considered to be stop codon, and one more you remember, triple U codes for phenylalanine. Now there are certain characteristic features, salient features of a codon. 6 salient features are there. This you have to remember. It is an important question in your board exam. First one is codon is a triplet. So how many codons are there? 4 to the power 3, 64 codons are there. In that 61 codon code for amino acids. But 3 codons are there that cannot code for any amino acid. They are called as stop codon, or it is called the nonsense codon. Which 3 are there? UAA, UAG and UGA. So codon is a triplet. One codon code for one amino acid. One codon code for one amino acid. So it is said to be unambiguous, or it is called as specific. No confusion at all. One codon code for one amino acid. So it is unambiguous or specific. But at the same time, some amino acids are coded by more than one codon. So the codon is said to be degenerative. Some amino acids are coded by more than one codon. So it is said to be degenerative. You have studied in the previous figure, you have gone through it. That is UUU is for phenylalanine, UUC is for phenylalanine. And then the codon is written in a continuous fashion without any punctuation mark. No comma, full stop, colon, semicolon, nothing. It is written in a continuous fashion. So codon is a triplet. 64 codons are there. 61 codon code for amino acid. 3 codons do not code for any amino acids. They are called stop codon. Example UAA, UAG, UGA. Second one codon code for one amino acid. So it is unambiguous and specific. Some amino acids are coded by more than one codon. So it is said to be degenerate. And a codon is always written in a continuous fashion without any punctuation mark. Now the codon is said to be universal. Why we are using the word universal? For example, you have studied triple U, code for which amino acid? Phenylalanine. In all the organism from bacteria to human, triple U codes for phenylalanine, but exceptional cases are of mitochondrial codon as well as protozoans. Except these two, in all other organisms, triple U codes for phenylalanine. So codon is said to be universal. And then AUG, just now you have come across, it is called the initiator codon. It is playing dual role. Dual role means a double role. One is it code for an amino acid methionine, and it is acting as which codon? Initiator codon. So these are the salient features of genetic codon. Now let us go to the previous one. You have studied about, that is you have studied about sickle cell anemia, and it is an example for point mutation you have studied. So what is a point mutation? Point mutation is of change in single base pair. So in case of sickle cell anemia, in the 6th position of beta globin chain, instead of glutamate or instead of glutamic acid, valine is present. So this is because of change in single base pair, and this result in a disorder. So how this type of changes occur? Let us learn from a particular example. So here a sentence is given to make you to understand. A sentence is given. Ram has a red cap. Try to understand every word consists of how many letters? 3 letters. So here every letter is equivalent to a nitrogenous base. So Ram is a genetic codon, Has is another codon, Red is a third codon, Cap is a fourth codon. Now let us insert. We are inserting a letter B in between Has and Red. So what is happening? We have inserted. So initially we have Ram has red cap, a meaningful sentence we have. Now what we did, one base, that is B is added between has and red. Now it has altered the reading framework. We are getting a meaningless sentence. So now next base we have added, that is I we are adding. So here two bases we have added. Again what we got? We got a meaningless statement. Now let us add the third base G. Here we are adding third base. So now we are getting a meaningful statement. Ram has big red. Got it now? Ram has big red cap. Right. So automatically we are getting a meaningful statement. So from this you can understand that when you are adding a single base, the reading framework changes. When you are adding two bases, the reading framework changes from the place of insertion. But when we add three bases are added, the reading frame get unaltered. So what we are getting after the third one added? Ram has big red cap. That means we are getting a meaningful statement. Right. Now in the same way, let us remove one by one. Only one and rearranging the statement. So which letter we are adding? Ram has big red cap. Right. Now we are getting a meaningful statement after the addition of which one? Three nitrogenous bases. In the same way, if you remove one base or two bases, we will be getting a meaningless statement. So the conclusion from the exercise is insertion or deletion of one or two bases change the reading frame from the point of insertion or deletion. But insertion or deletion of three or multiples of three insert or delete will not change the reading framework. The reading framework remains unaltered after the addition or deletion of three bases. Such type of mutation we call it as which type of mutation? Frame shift insertion. You have added three bases and you are getting a meaningful one, that is called as frame shift insertion. If you delete three bases, and that is called as frame shift deletion mutation. This is clear. When one base is added or deleted from the point of addition or deletion, the reading framework gets changed. That is point mutation. When you are adding two bases or deleting two bases, the framework get changed from the point of addition or deletion. But when you are adding 3 bases or multiples of 3 bases from the point of addition, or you are deleting 3 bases or multiples of 3 bases from the point of deletion, the framework remains unchanged or unaltered. That type of mutation, we call it as a frame shift insertion, or it is called frame shift deletion. Thank you all.